So this is going to be kind of a scattered conversation. Certain things are fairly obvious. Involuntary, right? You can voluntarily contract, right? Striated, branched, 1 to 2 nuclei. Okay. I'll explain. Not injunctions. Sorry. Gut junctions. Okay. So, if you have 2 cardiomyocytes coming together, look here. You see that zigzag line? So this is the intercalated disc. Am I clear? Does that make sense? It contains 2 things. It contains, well, I should add here, desmosomes and gut junctions. So desmosomes hold cells together because heart constantly contracts and relaxes. Does that make sense? So desmosomes basically do not allow cells to fall apart. Am I clear? Gut junctions allow cardiac cells to communicate, and this is crucially important. I'm going to ask an embarrassing question. Anyone remembers the muscle physiology, the skeletal muscle physiology pretty well? Kind of, you know. So remember, in a skeletal muscle contraction, you have a nerve ending that controls a skeletal muscle cell. In cardiac muscle, the level of synchronization should be insane. And think of it like, think what would be the load on the brain, or any structure for that matter, or any structure of CNS, to control cardiac contraction if that control would be exerted by a nervous system. That would be terrible, right? Control each and every cell in a highly synchronous manner. It's like, from the engineering standpoint, it's the nightmare. So instead, there are several cells in the heart that we're going to talk separately about. Several cells in the heart that set up the rhythm. Am I clear? And they communicate the contractile state to other cells via gap junctions. Does that make sense? So we usually say that cardiac muscle works as a functional syncytium. The word syncytium means like one cell, single cell. So functionally they work like one giant muscle cell. Is that clear? Does that make sense? Okay. Now, we got this. What is the mechanism of contraction? Mechanism of contraction is, no, we're going to talk about mechanism of contraction and then the action potential separately. Yeah, I think so. I think it would be better. So this is what happens. You've got central line, the M line. You've got thick filaments. Okay, so that's going to be myosin. You've got thin filaments. These thin filaments are attached to intercalated discs. Sorry, not intercalated discs, Z discs. Okay? And muscle cells form cross bridges, just like skeletal muscle. It's your classic skeletal muscle sarcomere, okay? Same story, same sarcomeres. Are you following me so far? And just like in a muscle cell, you have tropomyosin, and you have troponin. Okay? So, listen. T-tubules in the skeletal muscle is much less developed. So calcium connection. So let's put the steps down. So remember what the contraction starts with, right? It starts with the increased levels of calcium. So increased intracellular calcium. Now, where this calcium can come from? Outside or from sarcoplasmic reticulum. Does that make sense? It all starts with increased intracellular calcium. Calcium binds to troponin. Binding of calcium to troponin results to the shift of a tropomyosin away from actin and that results in the contraction. Are you following me? So listen up. I'm going to say it one more time. I'm going to go over it one more time with you folks. So we start with elevated levels of calcium in the cell. Does that make sense? Calcium in the cell binds to troponin. The binding to troponin results in a change in the tropomyosin shape. So actin becomes available, and it can interact with the cross bridges, with the myosin heads. Okay, so myosin heads form cross bridges with actin. Contraction is being initiated. Okay, it's really, for me, I always kind of mix up wording, so I should say that when tropomyosin changes the conformation, cross bridges are formed between myosin and actin. That's that. Does that make sense? So that's the contraction part. Now, during that process, cardiomyocytes are depolarized. And the depolarization and repolarization happens in several steps. And it all starts with the opening of our good old friends. So this is your resting membrane potential of a cardiomyocyte, and then look what happens. So first, cardiomyocyte gets depolarized. Okay, so this is depolarization. And this depolarization occurs due to the sodium voltage gated channels. Okay, does that make sense? So voltage gated channels open, resulting in depolarization of a cardiomyocyte. Now the big difference starts at the top of it, because at the top sodium channels close, but calcium voltage gated channels open, and this is called plateau. Okay? So why is that important? When cardiomyocyte is depolarized for an extended period of time, are you following me? Is depolarized for an extended period of time, it allows the contraction to be long. How long the contraction of a skeletal muscle? Milliseconds. How long the contraction of cardiac muscle? Not fully a second, but like half a second. Much longer. Does that make sense? So it allows your heart basically to do this and stay contracted for a while. Am I clear? Okay. And then finally, calcium channels, at some point they do close, and repolarization occurs due to the voltage-gated potassium channels. So one more time. During depolarization, sodium channels open, and sodium flows into the cell. Okay? Depolarizing it. When the cell is fully depolarized, sodium channels close here at the peak. And just like with skeletal muscle, they don't just close, they are being inhibited completely. So why it's important? The complete inhibition of sodium channels allows for absolute refractory period, which prevents tetanus. Okay, fuse tetanus, contraction that is like stays forever. At the peak here, where sodium channels closed, calcium channels open, calcium flows into the cell, maintaining depolarization. Does that make sense? Finally, calcium channels close, potassium channels stay open, and potassium starts to diffuse out of the cardiomyocyte, repolarizing it. Does that make sense? Are we clear? Any questions? You good? Now, if someone experiences myocardial infarction, oh yeah, I almost forgot. So what type of metabolism would cardiomyocytes have? What do you think? Your heart, is that like aerobic process or anaerobic process? What does it look like more? Huh? Aerobic, of course, yeah. You know, this is why we call like, aerobic exercise. Good for the heart, right? So, cardiac muscle does a lot of aerobic metabolism. Now, what happens if there is a myocardial infarction, MI? Blood flow to cardiomyocytes is blocked. So, if blood flow is blocked, do cardiomyocytes get oxygen? So, no oxygen available. Now, what type of metabolism do muscle cells switch to when there is no oxygen? Remember, after glycolysis, pyruvate can go into the transition step in Krebs cycle only if oxygen is available. If oxygen is not available, what happens to pyruvate? Fermentation. Your muscles can do fermentation just fine. So, fermentation. Fermentation results in the production, anyone remembers? Lactic acid. Lactic acid drops the pH in cardiomyocytes. Does that make sense? And that results mitochondrial damage and decreased calcium availability. Now, I was stupid to erase these key steps, but remember when we were talking about the process of, so a couple of things. Look, first of all, decreased cardiac availability, you're not going to have a proper plateau, right? So contractile force, will kind of be reduced. Does that make sense? Without calcium in the cell, you're not going to have a proper contraction period because calcium cannot bind. If there's no calcium, nothing binds to troponin. Does that make sense? And on top of that, calcium is crucial for the proper rhythm of contraction. If calcium is not available readily, the people with MI will develop arrhythmias. Does that make sense? We clear? So basically, this is why, like myocardial infarction by itself, it will, if it lasts long enough, it will result in a death of the cells. But what's important, it's not just the damage, it's the person can die of a heart, because if you think about it, heart attack per se, it's not like I can't fathom the size of a thrombus, the size of a plaque that will block like one large coronary artery. It's got to be some profoundly large thrombus. Does that make sense? But if a small part of the heart stops receiving oxygen properly, it can trigger the local arrhythmia that can become the overall arrhythmia of the entire organ. And in arrhythmia, ventricular fibrillation, that's it, heart stopped. That makes sense? So this is why, like people can die of some people get hospitalized, some people die right away. Questions?
Could you go, what makes the voltage-gated potassium channels open?
Oh, just when this reaches a certain level of, like, they open right about here. But here you have a tug of war. You have potassium going out, calcium going in. And for the time of plateau, calcium kind of wins. It maintains depolarization. But when the voltage reaches that particular point, it closes calcium channels, and the only channels that you have left are potassium ones. It's all voltage.
So are calcium channels during the plateau are slowly starting to close?
You ask? Kinda. Because when we say, there is a certain voltage and channels open, it's not as simple. Most channels open most of the time. Does that make sense? So like when potassium channels open here, they are open most of the time. Some of them close. Calcium channels, same story. They are open most of the time, and as voltage decreases, they are open less and less of the time. Calcium, yeah.
Okay.
And then at this point, they die. They just close.
Okay.
Does that make sense? And potassium channels remain open, and you have repolarization. So basically, the whole point of calcium channels being involved is to expand the time when cell is depolarized and is contracting. That makes sense? Questions? Okay.